Okay, because of a short that came up on my feed on YouTube about COVID-29, I decided to start doing some investigation, because on the video it talks about COVID-29, right? And it's saying that in the year 2033, so all these things are biblical, I think there's a correlation. The powers that be are naming things or choosing to name things after certain events. So they said that, just a minute. So, the fact that they're calling it a coronavirus 29, and that it's going to infect or wipe out many people in the year 2033 is what that video said. 33 is a Masonic number, and if I'm not mistaken, they claim that Christ came and his prophecy or his ministry started at the age of 29. So what are they really trying to tell us here with this virus? So let me read a lot of what this is saying here, okay? All right, it says these biomolecules could hold clues to why the virus is so infectious and to how to stop it. That didn't make sense. So this was written in April 1st, 2020 by somebody named Ala Katz Nelson. Okay. I don't know what this is. Let me try to click off that. There we go. Bunch of shit's popping up. Okay, it says a transmission electron microscope image shows the crown-like shape of SARS-CoV-2. Let's go look at it again. So if you don't think this is is biochemical warfare and created in lab and the names are naming it, it is very much a created thing. This is not something that just happened by chance. Just want to say that. It says scientists across the globe. Oh, by the way, the website is, here it is. Let me see. All right, here we go. It says scientists across the globe are gunning to understand the novel coronavirus called SARS-CoV-2 and what makes it so contagious and deadly. Several members of the coronavirus family infect humans. 4 cause the common cold, and 2 SARS-CoV and MERS-CoV have triggered dangerous epidemics. The novel coronavirus's closest kin is SARS-CoV, or SARS-CoV, which jump species from bats to civets to humans. I don't believe that. To cause the severe acute respiratory syndrome SARS epidemic in 2002-2003. That SARS outbreak infected over 8,000 people. The current coronavirus pandemic has infected more than 880,000 people and killed more than 44,000. As of April 1st, according to the data from Johns Hopkins University. From a molecular perspective, figuring out why the virus is so much more transmissible than past viruses is where we should be looking right now, says Robert Kirchdorfer, a structural biologist at the University of Wisconsin-Madison, who studies how coronaviruses fuse with host cells. While people infected with the early 2000 SARS virus shows severe symptoms almost right away, people infected with SARS-CoV-2 can spread the virus before they show symptoms or why they are just mildly sick. A study published in Nature report that patients shed virus most efficiently in the first weeks of illness when their symptoms are mild. 2020 DOI, and then it has a number, 10.1038/S41586/020/2196/X. The U.S. Centers of Disease Control and Prevention now estimates that 25% of people infected with the virus show no symptoms. That gives it an advantage to spread, says Melanie Ott, a virologist of the University of California, San Francisco, and a member of the Quantitative Biosciences Institute's COVID Consortium. There must be something in this virus's biological makeup that facilitates this silent transmission and that makes its effect on its human host so variable. But researchers don't yet know what it is. The RNA genome of SARS-CoV-2 has 29,811 nucleotides encoded for 29 proteins. Though one may not get expressed, studying these different components of the virus as well as how they interact with our cells is already yielding some clues, but much remains to be learned. All right, let's go on. It says SARS-CoV-2 has 4 structural proteins. Top, the E and the M proteins, which form the viral envelope, and the N protein, detail not shown, which binds to the virus's RNA genome, and the S protein, which binds to the human receptors. Sorry, gotta scratch my nose. The viral genome consists of more than 29,000 bases and encoded and encodes 29 proteins. Bottom, the non-structural protein gets expressed as 2 long polypeptides, the longer of which gets chopped up by the virus's main protease. And this group of proteins includes the main protease, NSP5, and RNA polymerase, I don't know if I'm saying it right, MERASE, NSP12. The spike coronaviruses are named for the crown of protein spikes covering their outer membrane surface. Early work on the novel coronavirus has focused on these spike proteins, also called S proteins, because they are the key that the virus uses to enter the host cells. In both SARS-CoV and SARS-CoV-2, the S protein binds to the receptor called angiogenesin, converting enzyme 2, ACE2, to hack its way into host cells. At the amino acid level, the spike proteins of SARS-CoV-2 are about 80% identical to those of SARS-CoV. The residues which interact with ACE2 are conserved compared to the first SARS, but the residues in between are different, and there are also some insertions, says Rolf Hillingerfield, a structural biologist who studies coronaviruses at the University of Lübeck. Studies so far suggest that the new virus spikes proteins binds to ACE2 significantly more strongly than those of SARS-CoV. That is probably one of the reasons it spreads more easily and is more infectious, Hilgerfield says. From a molecule perspective, figuring out why the virus is so much more transmissible than the past viruses where we should be looking right now, says Robert Kerdorfer, the structural biologist of University of Wisconsin. Let's see. A non-peer review reprinted posted on medRxiv on March 27 reports that ACE2 is expressed especially strongly in the lungs of people with lung disease of 2020 DOI, and then it's 10.1101/2020.003.20040261. That observation may partly explain how these underlying conditions make people more susceptible to infection by the novel coronavirus. Stronger binding with the ACE2 isn't the only clue to the virus's power. S proteins are made up of two segments, S1 and S2, that must be cleaved at two sites to expose a peptide that initiates fusion with the ACE2 on a host cell. And I'm going to go into ACE2 because that seems to be a major factor to this. A mutation in the SARS-CoV-2S protein allows an enzyme called furin, which is made by many types of human cells, to do the first cut. This ability to get human enzymes to do its work primers the virus for fusing with H2, providing another possible explanation for this virus infectiousness, says Carolyn Machamber, a cell biologist at Johns Hopkins University who studied the basic biology of coronaviruses. It can get this first clip before it even comes into contact with the receptor. I'm sorry, but I think keywords to look for are ACE2 and where is it? Hold on. This ability to get human... Hold on. Furin. So I think some of the keywords here are furin. Because furin doesn't... I don't know where this is from interspecies thing can happen. If it's saying this is furin, which is made by many types of human cells. I mean, do bat cells have furin? I don't know. So how is this connecting? So I want to research that. It says researchers are working on identifying the best way to interfere with the S protein's interactions with ACE2. ACE2 is present in many organs throughout the body, and interfering with it may have side effects. So researchers want to avoid hitting the receptors and are instead developing antibodies or peptides that bind and disable specific segments of the S protein. The other 28 of the 29 SARS-CoV-2 proteins, 4 make up the virus's actual structure, including the S protein. One group of the other 25 coronavirus proteins regulates how the virus assembles copies of itself and how it sneaks past the host immune system. These so-called non-structural proteins are expressed as 2 huge polyproteins that are then cleaved into 16 smaller proteins. An enzyme called the main protease, which performs 11 of these cleavages, is also a high promising drug target. Hillinger Field and his colleagues recently reported the structure of the main protease and identified an inhibitor that can block it. Many of the virus's non-structural proteins are still poorly understood. I think we are just going to have to brute force and probably piece through the genome and study a lot of these proteins individually, says Anthony Fair, a biologist at the University of Kansas. These studies will help scientists both understand the underlying biochemistry that gives SARS-CoV-2 its nasty kick and identify other ways to figure the virus. Fair's lab work on a non-structural protein called NSP3, a component of which blocks the host's efforts to fight off the virus. What? Called PARPs. This protein shuts down the host enzymes called PARPs, which prevents viruses from replicating and interferes with the cellular calls for the release of the virus-fighting immune proteins called interferons. Whoa, where is it? This protein could be a target, he says. The virus would be dead in the water if it didn't have a way to counter the interferons response. Okay, we're gonna have to look those up. Those are keywords. The third group of proteins in the novel coronavirus are accessible proteins. Let's see. Meanwhile, other labs are rushing to explore how differences in the immune system response might affect how severely ill an infected person may get. For example, Oates' lab is beginning to infect organoids, which are models of lungs and other organs made of complex cell structures, with the virus to watch how infections proceed. The scientific community has come together in an unprecedented way, Oates says. Everybody basically has shifted their attention to the virus and is looking for ways to contribute meaningfully. Okay, so I'm going to upload this because it's 20 minutes long, and then hopefully it'll get uploaded, and then I'm going to research these other keywords.